I'm going to be talking about alterations that occur in later stage sepsis, that in particular dysregulated leukocyte trafficking, that we feel contributes, at least in part, to the immunosuppressed status that physicians see in septic patients as they are being treated in the hospital. So patients that have a bacterial infection that proceeds to a systemic infection, they have an overblown inflammatory response that can contribute down the road to multiple organ dysfunction, and in some cases the patients don't survive. But there are a number of individuals in this situation who, with aggressive therapy, antibiotics, and improved clinical care, that they're clinically stabilized. But not all of these individuals who are clinically stabilized go on to recover and leave the hospital. There's a significant proportion of these patients who develop secondary complications. And while they can recover and again be discharged from the hospital, there is a significant death that comes from these secondary complications. So what are the secondary complications, and what are the causes for them? Two of the main complications, the first is an increased susceptibility to secondary lung infections, in particular hospital-acquired pneumonias in patients who are septic. This complicates treatment of the primary infection in these individuals, lengthens the time they're in the hospital, and really decreases the survival rates of these individuals. Another major complication that septic patients have to deal with is that they have impaired wound healing, in particular with abdominal incisions. So what is it that contributes to these complications? Well, the fighting off secondary lung infections and healing wounds, these two processes really require the ability to recruit inflammatory cells to, say, to the lung to fight a lung infection, or to the wound site. And therefore, it's potentially possible that if the ability to recruit cells was impaired in septic patients, that this would interfere with their ability to fight infections and heal their wounds. And so what are the factors that allow for the recruitment of cells? There are bacterial products like formulated peptides, also many endogenous host factors, components of the complement cascade, neuropeptides, components of the cyclooxygenase pathway. But also there are cytokines, and in particular a family of proteins called chemokines that really are major players in recruiting cells to sites of infection and also to wound sites. It's a large family of proteins that can be actually subdivided. Some of the family members are actually constitutively produced and actually play more of a role in homeostatic processes, while there are other chemokines like interleukin-8, which we've heard a lot about today, that are not expressed constitutively, are only expressed when one comes in contact with an activating molecule like a bacterium. And it's these molecules that need to be expressed and to recruit cells to sites of secondary infection and also wound healing. And just like with all the other processes, HMGB1, MIF, balanced chemokine responses are essential for successful resolution of infection. If you have the appropriate amount of receptors that bind these ligands and you have appropriate amount of the ligands themselves, you get protection. If you have uncontrolled release of these early in sepsis, for instance, you have the infiltration of large numbers of inflammatory cells, activation of those inflammatory cells, and the resulting pathology. But we and others have shown that if you knock out these molecules or their receptors, either with antibody neutralization or in knockout animals, that you get an inadequate host response and you can't fend off infections and you can't heal wounds. Sounds very similar to what you see in patients with sepsis.